my dear students, welcome to Botany class. How are you everybody? I hope you all are well and safe. Today we are going to discuss about some important topic from chapter 12, that is heredity in organism and evolution. Now let's see the first topic, that is gene. The unit of controlling all the visible and invisible signs and characters of organism is called gene. Gene is the smallest chemical unit which bears heredity characters. In other words, any part of DNA molecule that can take part in mutation is called gene, or gene is a part of DNA which bears the genetical information of living organism. This one is a DNA, so we can say that a particular segment of DNA is called gene. Okay everybody, and the location of gene on the chromosome is known as locus. Generally, one pair of gene is responsible for one characters. In some cases, more than one gene unitedly responsible for one character. Gene is formed by replication of old DNA. So, the new gene are produced from old one. As a result, gene is unchanged from generation to generation. From different research, it is clear that a gene is the perfect master of heredity. By taking necessary signals from DNA, RNA enters into the ribosome of the cytoplasm, and according to this signal, protein is synthesized. Here, the production of mRNA from DNA is called transcription, and production of protein from the mRNA is called translation. This protein determines the nature and speed of biological reaction, and from that, the characteristics of living organism are determined with respect to the environment. These characteristics include everything from structure to the behavior of any living organism. It is observed that the word gene comes from the Greek word genus, which means to burn. It is observed that all the body cells contain same type of chromosome with same type of gene, but the functions of the different type of cells are quite different. Why it is happened? This is due to enzymes. Gene is the smallest part of DNA. On the other hand, enzymes are protein. Enzymes enhance the biological reaction but remain unchanged after the reaction. These enzymes are produced by the influence of gene. Specific enzymes control the activities of specific sets of genes. So the gene and enzyme are interrelated and carry out all the functions in a cell. In other words, it can be said that the activity of gene is influenced by the enzyme. Therefore, they are interdependent with each other. Gregor Mendel in 1860 for the first time observed in his experiment with pea plants that heredity characters are transmitted from the parents to the offspring in a certain rules, which is controlled by some particles called as factors. In 1909, Johnson named these Mendelian factors as gene. So, according to the opinion of the father of heredity, Gregor Johann Mendel, each characteristics of organism is regulated by a pairs of factor. That means gene. In this figure, this person is a Gregor Mendel. Gregor Mendel was an Australian monk who discovered the basic principles of heredity through experiment in his garden. Mendel's observations become the foundation of modern genetics and the study of heredity, and he is widely considered as pioneer in the field of genetics. Gregor Mendel is known as the father of genetics for his valuable works in genetics. Now let's see the experiment of Mendelian. To see how the characters or traits of parents are expressed in the next generation, scientist Gregor Mendel did some experiments with pea plants in 1866 and discovered a valuable theory of genetics. Mendel took a tall and a short pea plants and transferred pollen grain of a tall plants into the stigma of a short plants. Here in the figure, this one is a tall plants and this one is a short plants. In the same way, he transferred the pollen grains of a short plants into the same stigma of a tall plants. This resulted in reproduction. He made sure that the tall dwarf other pollen grain would not appear. In his first experiment, Mendel made cross between tall plant and with the dwarf pea plant. Here, Mendel crossed between two plants, tall plants and dwarf plants. In this figure, the capital T gene is responsible for the tall plants and small t is responsible for the dwarf plants. The genotype of tall plants will be double T, capital double T, that means capital T, and the genotype of dwarf plant will be small t, these two. Therefore, the gamete of tall plants will be T and gamete of dwarf plant will be small t. As a result of cross, the genotype of F1 plants will be capital T and small t. Here, the genotype. This one is a genotype. Genotype will be capital T and small t. This one is capital T small t is a heterozygous tall. That means one from father and one from mother. All the F1 plants, that means first one generation, here F1 means first filial generation. All the F1 plants, the phenotypically will be tall because of T, here is T gamete is a dominant. Next time, when the self cross is made between the F1 generation, then two types of gamete, that means here two types of gamete, one is capital T and another one is small t. In the same way, here one is small t and another one is capital T. When these gametes are crossed again in F2 generation, this one is a F2 generation, 1/4 will be 1/4, that means this one will be heterozygous tall. Sorry, this capital T is a homozygous tall and half will be the heterozygous tall and 1/4 will be homozygous dwarf. That is 25% of plants will be homozygous tall, 50% heterozygous tall and 20% homozygous dwarf. The phenotypic generation of the tall and dwarf will be 3:1 and the genotype will be 1 homozygous tall, 2 heterozygous tall and 1 homozygous dwarf. Therefore, the genotypic ratio is 1:2:1. 1:2:1. It can be revealed that due to the presence of a dominant gene in the genotype, the dominant character will be expressed, and absence of dominant gene, the recessive character will be expressed. This theory of Mendel is applied in breeding of plants and animals. The controlled sexual reproduction is done in plants and animals to bring about desired characteristics. As a result, a generation with various characteristics are produced. From this generation, desired characters are selected for breeding to get plants and animals that have desired characteristics. At present, this theory is applied legally for the production of improved variety of crops. Now let's see the replication of DNA. What is DNA replication? The process by which a new DNA helix is formed from the old DNA double helix is known as DNA replication. Let's see the figure. Here is DNA replication takes place. This process takes place at the interphase stage of cell division. The replication mechanism proposed by Watson and Crick is quite simple. In the newly formed daughter DNA molecule, one strand will be old and another will be new. This is known as semi-conservative of replication model. About 20 proteins and some enzymes are related in this process. These components are known as DNA replicase. At first, the hydrogen bond in between two nitrogenous base breaks down, thus double helix DNA unwound to form a single strand. Here is single strand. Now, each single strand is used as template on which complementary helix is formed. The materials of the new DNA helix are available in protoplasm of the cell. The enzyme DNA polymerase and magnesium ion plays an important role in this process. When the double strand DNA is unwound up to a point of a Y-shaped structure. Here, Y-shaped structure is developed, which is referred as replication fork. The enzyme DNA polymerase can polymerize the nucleotide only in the 5' to 3' direction. Adenine connected with thymine and cytosine is always connected with guanine. The source fragments of DNA are joined together by hydrogen bond and enzyme DNA ligase. This complementary two-strand DNA is formed in antiparallel orientation. Now let's see the genetic disorder. Genetic disorder is a disease that is caused by an abnormality in an individual's DNA. Abnormalities can range from a small mutation in DNA or addition or subtraction of an entire chromosome or set of chromosomes. Here, mutation means some changes occurs in DNA. Color blindness is a sex-linked disorder. In some genetic disease, a mutation occurred in the gene of sex chromosome. This type of disorder are called sex-linked disorder. Here, color blindness is a sex-linked disorder. Color blindness occurs when you are unable to see colors in a normal way. It is known as a color deficiency. Color blindness often happens when someone cannot distinguish between certain colors. This usually happens between green and red, and occasionally blue and yellow. In this picture, a color-blind man will see the number 21 inside the figure. A color-blind man will see the number 21, whereas a healthy man will see it 74. Color blindness is a condition when someone cannot properly identify colors. To identify colors, we have pigment in our optical nerve cells. If color blind, the patient is deficient of some identifying pigments in their optical nerve. If someone lacks a single pigment, then he would not able to differentiate red and green colors. This is the universal problem of color blindness. If lacking more than one pigment beside red and green, the patient cannot differentiate the color blue and yellow. 1 man out of 10 is color blind. In comparison, a few number of women suffer from this problem. Now let's see the autosomal recessive disorder. If the disorder is autosomal recessive, there should be transfer of both affected genes from both the parents to causes the disease. If gene from only one parent is transferred, then the child becomes a carrier but does not get the disease. Thalassemia is the example of autosomal recessive disorder because in the autosome some changes occurs, and when some changes occurs in autosome, thalassemia is created. Thalassemia is an inherited blood disorder caused when the body does not make enough of a protein called hemoglobin, an important part of red blood cell. Thalassemia is a disease of abnormal state of red blood cell. Because of this disease, red blood cell disintegrated. The patient suffers from anemia. This disease genetically passed from generation to generation. Autosomal recessive disorder means when both father and mother are the carrier or both are the patients of thalassemia. Only then does it dominates in the offspring. When the marriage of held between paternal or maternal cousin or between close relatives, there is a higher probability of giving birth to a child with thalassemia. Thalassemia are of two types depending on the inherited gene. That means major thalassemia and minor thalassemia. Major thalassemia means the victim baby obtains genes from both parents, and in case of minor thalassemia, a baby obtains genes either from mother or from the father. This type of body does not express any sign of thalassemia but function as a carrier of thalassemia gene. On the other hand, there are two types of thalassemia due to two types of gene disintegration, that is alpha thalassemia and beta thalassemia. Alpha thalassemia is caused when alpha globulin production is absent or defective in the body. And this type of disease is alpha thalassemia. And beta thalassemia, in the same way, when the beta globulin production is stopped or defective, then beta thalassemia occurs. Everybody see the figure. Here father is an unaffected carrier father and mother is an unaffected. And here capital R is a healthy gene and small r is a thalassemia gene. Two types of gamete is formed from both parents. When cross occurs between two types of gamete, it is seen that out of 4, 1 is unaffected child and 2 are carrier but unaffected child, but 1 is affected child. This one baby can carry the thalassemia gene, that means both are capital small r. Okay, today up to this everybody. Thank you.